Couple quick reminders. I think everyone has a copy of the lab manual now after kind of some of the bookstore issues. If you don't, please let me know. There should have been more than enough copies left over there, but if you don't have a copy or head over get one, just talk to me after class here, just so that I make sure that that's kind of all taken care of. With that, just for next week, we kind of have the same general thing you guys can be expecting next week for assignments. Just what's coming up in the class on Tuesday, we'll have our next lab activity. Make sure you're reading through the lab activity before you come to lab class and then completing the pre-lab quiz number 3 that's on Brightspace. So that is, you have a pre-lab quiz that's due on Tuesday. Tuesday's lab is going to be a population genetics lab. So we're going to be talking about evolution today, kind of finishing up a few, if we don't finish up with the evolution lecture set today, we'll come very, very close. And then on Monday's lecture and Wednesday's lecture, we're going to be talking about population genetics. For Tuesday's lab is also going to be about population genetics, but it's kind of the first part of the lab. We're not going to be talking directly about population genetics in class on Tuesday in lab, but we're going to be starting the first part of a 2-week experiment on that. So we're going to be talking mostly about some techniques and doing some techniques to start the data collection process on the lab. We're going to be tracking and doing population genetics assignment on a particular population, which is our class. So I'm actually going to collect genetic data from yourselves. So we're going to actually on Tuesday harvest some DNA, isolate DNA from some of your cheek cells. We're going to extract that DNA, and then we're going to actually amplify up one of the genes in your DNA called the BV92 gene. And then we're going to basically genotype you for that BV92 gene. So we're going to do a process. We're just going to isolate the DNA, do something called PCR, polymerase chain reaction. So I'll teach all about that. And then we're going to go through that and collect some of that data. The PCR itself takes a couple hours. So we're not going to have time to do everything next week. So we're going to do the PCR, get it set up. And then we're going to let that run. I'm going to keep those until the following week, and then we're going to collect the data on that using some other techniques. And then we're going to analyze it using some population genetics that we're going to be talking about on Monday's lecture and Wednesday's lecture. Then the following after. So this is going to be the first part of a 2-week lab, and we're also going to start talking about some statistics as part of that lab in a couple of weeks when we go through the chi-squared statistical test. So several different things going along in lab and lecture kind of simultaneously to complement one another. So that's Tuesday. The other thing we're going to be doing next week is then, remember you'll have another Brightspace quiz on Wednesday next week. So pre-lab on Tuesday due, Brightspace lecture quiz on Wednesday after class. Same format as we've been doing. Also make sure you're checking the feedback on those quizzes. So you should be able to go in and see, again, which questions you got right, wrong, but you might have feedback associated with every single question on those to kind of help through the thought process and what was the right answer, and also kind of my versions of the answers that were kind of, I was looking for that in many cases. Yeah, I would also suggest you guys take a look with that, even if you got the question right. Sometimes with the grading, I am usually fairly lenient with the grading as long as you were on the right track. But I do find that sometimes the phrasing that some of you guys have been using on some of the questions, while you might have got full credit, I would kind of pause and look at it for a while. I do find sometimes the descriptions are just a little bit off. And sometimes if they're a little bit off one day, they're a little bit more off the next day, a little bit more off the next day. And then by the time the exam rolls around, you've kind of, everybody ever played that game of telephone in school where you pass the messages back and forth and the message gets messed up over time? That's what tends to happen sometimes with, by the time the exam rolls around. A small little mistake gets modified into, evolves in essence, into a bigger mistake. So make sure you're going back and checking even feedback on questions you might have gotten right is a suggestion there. And again, if you have any questions whatsoever, about any of that feedback or why you got something wrong, feel free to send me an email, stop by my office, check through, but it's always good to get some extra help. And don't wait until right before the exam when you might need, it usually just works best if it's kind of involved in. All right, otherwise today, we're gonna keep on talking about evolution. And we left off talking about evidence for evolution. And so I went through the example of, for instance, the guppies in the pond and a specific experiment that was done with some of those. I did want to also just bring up this idea that there's a lot of examples of human evolution. So for instance, the lactose, some people can tolerate eating lactose in milk and some people cannot. It turns out back in ancestral human populations, if you go back about 8,000 years ago, all humans were lactose intolerant. It actually turns out that it started out that all humans were lactose intolerant. If you ate milk as an adult, it would create gas and tummy issues and all that stuff like that. And then it turns out what ends up happening is infants have an enzyme that can process lactose in things like breast milk. But in adults, that enzyme turns off permanently. But about 7,500 years ago in the Hungary regions where they think it is, in Europe, is that there was a mutation that took that lactose processing enzyme that was only on in infants. It basically made it so that it was also stayed on in adults, and the adults could eat the milk properly then as well. And that mutation has spread out of then southeastern Europe into all around the world, in essence. And that was just because that was milk and cheese food sources became kind of with the advent of farming and farming practices, people started consuming milk or cheese or other kind of fermented products, and being able to eat lactose was an advantage. And so that mutation has spread. There's things like the sickle cell allele malarial resistance that we've talked about. The sickle cell allele is more prevalent in areas where malaria is, and provides resistance to malaria. But that sickle cell allele, we also see simultaneously that its frequency goes way down in areas that malaria is absent, for instance. So we can see those inheritance patterns and those changes based upon that. Blue eyes in humans actually only developed about 6,000 years ago. We can kind of track these in genetic markers from archaeological digs and things like that. And they can track what genes, the OCA2 gene that resulted in that. There is things like the high altitude adaptations, Tibetan versus Han Chinese populations. Used to be the Han Chinese and Tibetan populations were the same population about 5,000 years ago, but then they split and the Tibetan population moved up to high altitudes, living up high in the mountains, whereas the Han Chinese stayed in more low-lying areas. While they shared a common ancestral population genetic background, the Han Chinese kind of just stayed with their similar genetic background, while the Tibetan population acquired a bunch of high altitude mutations that let them process oxygen better, where oxygen is less prevalent at those high altitudes, for instance. Wisdom teeth have been gradually disappearing in human populations over time. And now some people either are missing some wisdom teeth or even don't even have wisdom teeth in the first place. So for instance, my wife doesn't have wisdom teeth. Doesn't have to worry about that. Dentist visit, for instance. But there are some individuals that don't even have wisdom teeth anymore. And so, let's see. Alcohol flush in Asian populations. Turns out there are some genetic markers in Asians that make it so that alcohol hits them more strongly. And so I don't know if you've ever seen someone that gets really, really red-faced, flushed after even consuming only a little bit of alcohol. It's actually a reaction. They have a genetic marker that makes it so they cannot process alcohol very well. And they've actually evolved a mutation also that makes them then strongly react to alcohol so that they avoid it then as well, and it's related to that. And then also human brains have been gradually shrinking since about 20,000 years ago. Human brains have gotten smaller by about the size of a tennis ball based upon archaeological records than in fossil records. And so the granule brain has actually gradually been shrinking for about the past 20,000 years. And these are all like documented changes over time in human populations. And there's a whole bunch of others. I'm just giving you a select one. I just have some links to a few articles about human evolution and examples then of human evolution if you guys are interested.
How is the human brains evolution?
So it's actually, they see brain shrinkage in all domesticated animals. So if you look at dogs over time that have been domesticated by humans, you see their brain shrink over time. Cows, pigs, chickens, all of that, you see brain shrinkage over time in domesticated animals. And I would actually lump, strangely enough, humans in many ways could be considered domesticated. I know that's kind of a horrible way of stating it, but there is agriculture. There is better building and shelter construction. And so what ends up happening is humans don't have to worry about predators, kind of top of the food chain. And so what you've lost is sections of the brain often involved in a lot of the vision stuff and maybe recognizing predators. You've lost some portions of the brain that are more involved in kind of predator. It seems like they're like more developed in animals that have to avoid predators, for instance. So if you actually have changes in the brain, but specifically changes in the brain that are fairly energy intensive, but then simultaneously don't really provide as much advantage anymore in our current natural selective pressures. Does that answer the question?
Yeah.
Domesticated animal way is not the right way to phrase that, but it's the one that grabs you guys' attention. Okay. Antibiotic resistance in bacteria is another example of evolution in action that we can see. So for instance, this is year versus methicillin-resistant Staphylococcus aureus infections. So Staphylococcus aureus is a bacteria that causes staph infections, mostly skin infections, but also very, very rarely necrotizing fasciitis, and which you guys might know better as flesh-eating bacteria, which unfortunately, I will warn you, I have kind of a nasty picture on the next slide regarding that. And so with MRSA infections, there's a lot of different bacteria that can cause necrotizing fasciitis. Staph is just one example of a bacteria that can cause necrotizing fasciitis. But it's been also increasingly resistant to methicillin. So this is annual hospital admissions with methicillin-resistant staph infections. And actually, they've gotten so common now, my data's a little old here, but they've gotten so common, they've stopped using methicillin antibiotics to treat staph infections. It just doesn't work anymore, that particular antibiotic. They've had to switch over to other antibiotics to treat staph infections. And then this is actually a percent resistant strains to antibiotics in human fecal matter versus then antibiotic usage in tons. And typically, the more you use an antibiotic, the faster and the more you get antibiotic resistant bacteria to that antibiotic is just what this graph is kind of showing. So antibiotics that are used quite common like ampicillin, kanamycin, streptomycin, tetracycline, these might be all ones that maybe even you guys have used to treat a disease before. Typically, the more they're used, the more often we see antibiotic resistant strains. For instance, tetracycline is pretty heavily used. And if you survey human fecal strains, you actually find about over 80% of them are resistant to tetracycline. So the more you use the antibiotic, the more that you get this. And this is just a really strong selective pressure. What do antibiotics do? They kill bacteria. That's a strong selective pressure, killing. Okay, and the more often we use it, the successful strains that want to make it through are ones that have some form of resistance to it. And because they're the only ones that live, they recolonize and then they become much more common over time. And we see that developing. And again, this is where I say I had kind of some nastier slides. There are pictures here. Methicillin-resistant Staphylococcus aureus infections are particularly becoming more antibiotic resistant. They usually just cause like pimples, abscesses, rash, is what they usually just cause. Very, very rarely, they're associated with necrotizing fasciitis or flesh-eating bacteria. So for instance, this is a patient's thigh. And this is kind of severe necrosis on the leg. Typically, the only way to treat that is by surgical debridement. They cut away all of the infected tissue, and sometimes that can just be cutting away the infected tissues in that area, but other times it requires full amputations of limbs, say, for instance. Yeah, and
How does one come in contact with infection?
well, if you want to know the disturbing answer, that's really actually not that disturbing. First off, 33% of you in the class right now have Staphylococcus aureus on your skin or in your nose. This is something we're going to talk a little bit about. Staphylococcus aureus and how you actually even define bacterial species in general or other bacterial species. It's really hard. There is so many different kinds of bacteria out there that it's actually hard to define what a bacterial species is. So when I say Staphylococcus aureus, there's Staph aureus, but there's a whole bunch of substrains of Staphylococcus aureus. Some of the Staphylococcus aureus is commensal, and we're going to talk about commensal relationships later in the class, but that just means that it is neutral for us, but the bacteria gets benefit from just like eating our skin secretions and things like that. It doesn't cause us any harm. The bacteria gets a nice place to live. There are some versions of Staphylococcus aureus that are pathogenic, that can cause disease, but are not antibiotic resistant. There's some versions of Staphylococcus aureus that are disease causing and also antibiotic resistant. And you basically just have to come in contact with one of the kind of bad varieties of staph. And it's floating around all sorts of places in the environment and on other people. Staph infections, for instance, are really bad about spreading in like between wrestlers and things, but it usually is about skin to skin contact with somebody else. Or yeah. And that's why it's particularly bad, like spreading in between wrestlers. All right, antibiotic resistance is particularly bad for a couple different reasons. There are actually some resistant pathogens of things like Enterococcus faecium, Mycobacterium tuberculosis, Pseudomonas aeruginosa, Candida albicans, which is actually a fungus. But there are some varieties of these pathogens, substrains of these pathogens that are resistant to all antibiotics that we know of and use to treat disease. So they're not just resistant to one kind of antibiotic. There's some versions of these pathogens circulating around that are resistant to multiple antibiotics simultaneously, including many of the ones that we treat disease now. And so these can sometimes create untreatable infections. That's a big problem, especially because this is the human growth curve. From about 1500 up to kind of present day. What you kind of see with the human growth curve, it's what's called biphasic. We have one rate of growth here, and the slope of my red line is the rate of growth in kind of this portion. But this is exponential growth. We're going to talk a little bit about that later on in the semester. But human populations really started growing really fast around like 1600. Their rate of growth increased. This was partially due to things like improved agriculture, but also they started using sewer systems. Simple sewer systems in cities, so you had improved sanitation that lowered disease spread, for instance. And so it's a few different things there, but starting in the mid-1900s or early to mid-1900s, we can actually see we have a faster rate of growth. It became kind of almost even more exponential in some ways. And so why is it, do you think we had a faster rate of growth in, say, the 1930s to 1950s that started? So medicine, and there were specifically two types of kind of medical advancements about that time. Penicillin, and so penicillin was first went into widespread use in 1942. And so they started using the first antibiotics. Antibiotics started to be used to treat disease, and then what that did is it lowered death rates. It's lower death rates substantially, especially in children. For instance, then, and that's going to accelerate the growth rate. So antibiotics was big there. Antibiotics has led to surgery, actually. Surgery used to be very, very, very risky because patients got infections. And now with antibiotics, it's made surgeries much safer and a much more common remedy for things than it was previously. Vaccines in general. So the other, it's mostly antibiotics and vaccines. And so with vaccination, that came about the same time, but that actually helps with disease prevention then as well. Anybody happen to know what the #1 cause of death is currently in the US?
Accidents.
For failure.
Heart disease, actually. #2. Cancer. #3 is COVID at the moment still. The #1 cause of death used to be, prior to say the 1900s, infectious disease. And so the vaccination has put that way down on the list. Antibiotics have put that way down on the list then as well. But again, bacterial populations are becoming antibiotic resistant, so that's going to create a lot of problems. This one takes a little bit to go through, but Richard Linsky has been doing experiments with Escherichia coli bacteria. Linsky's group has been studying the Escherichia coli strain since 1988. Actually, what they did is they took an ancestral population of E. coli. They divided it up into 12 different flasks. Okay. And so they took that population and they separated it into 12 different subpopulations. And then what they've been doing is they've been tracking that if they transfer it daily into new media, they've just been tracking what changes about it since 1988. And that is over 50,000 generations of Escherichia coli, because it goes through about, on average, about 40 to 50 generations per day. Bacteria grow really quick. And during that time, they've basically just seen, they took this sample of E. coli and they've been tracking it. They've been able to track things like increases in cell size over time, because they're growing them in very nutrient-rich environments. And cell size or surface area to volume ratios help with nutrient uptake. But because they're growing them in very nutrient-rich environments, cell size and surface area to volume ratios are not that important. So they're really always having a lot of nutrients around, and so surface area to volume ratios aren't important, and they got a little bit bigger over time. They also, in 2 of the 12 cultures, evolved the ability to eat citrate. So Escherichia coli typically does not have any enzymes for processing citrate. In 2 of the 12 cultures they've been growing, they evolved and changed so that they could eat citrate, for instance. And actually they mapped it to 2 separate mutations that happened independently of one another. So it's actually was a modification in one enzyme and then a second modification in a different enzyme that allowed them to eat citrate, for instance. They actually freeze the E. coli populations down. They store them in a -80 freezer. They do that daily, so they have a living fossil record. They can go back to any particular day, recover the E. coli, sequence its genome, do things, and say, oh, this was its genetics on May 6th of 1999. So they can go back and take a look at this, at the genetic changes that have mapped up over time. Okay. All right. So those are some direct experimental evidence of evolution. We also have other forms of evidence for evolution. So another form of evidence for evolution is homology. Homology is trait similarity or similarities that result from common ancestry. And when you have these trait similarities, one example of trait similarities are then what are called homologous structures. These are anatomical resemblances, anatomical anatomy similarities that represent variations or modified versions of a structure from a common ancestor. So what we have here is, say you have several different species that are descended from a common ancestor, they're gonna have similarly shaped structures in them. And that would be homologous structures. And an example of this would be arm structure in mammals. So humans are mammals, cats are mammals, whales are mammals, bats are mammals. Okay? And if you look at their forearms, if you look at their arms, their front, they have the same bone layout. So you have a humerus marked in purple, you have a radius, ulna, you have carpals, metacarpals, and the phalanges. And you have that same structure and layout in cats. Again, humerus, radius, ulna, carpals, metacarpals, phalanges. They're in the same layout, they're just slightly modified versions of that. But you can get to some more substantial modifications. If you look at whales, well, they don't really have arms, their arms have modified into flippers instead over time. Whales' ancestor was a land-based mammal. The ancestor of whales was a land-based mammal, but it started living in intertidal zones and eventually it moved fully back into the ocean. And its arm changed into a flipper over its evolutionary history. But again, it still has a humerus, radius, ulna, carpals, metacarpals, and phalanges. They just have been modified. They are in the same basic layout, but some of, say, the phalange bones have been modified and elongated. And then you can actually see that elongation even more so in bats. Wings of bats, again, is a modified arm, but it's got a radius, humerus, radius, ulna, carpals, metacarpals, but you can just see their metacarpals and their phalanges have just elongated quite a bit to hold up their wing structure. But you have the same basic bones. You really have honestly mostly the same number of bones in the same layout. It's just the bones have a different kind of overall modified shape to support some of these different structures. So those are an example of homologous structures. Questions about that? You can have homology in homologous structures. You can have, we see homology in embryonic development. So for instance, this is a embryo from a chicken versus a human embryo. And if you look at them, especially early on during embryonic development, they have a lot of the same structures and look fairly generally identical. So you have some things like pharyngeal pouches. Those are going to become things like the larynx, but they look like gills at this point in the embryonic development a lot, because remember, I already had said that the larynx, the voice box, is a modified ancestry, is a modified gill. So you have these pharyngeal pouches, and human and developing embryos start off with a tail, for instance, but then they lose it over the course of the embryonic development. How do they what?
How do we look at those? Because do we look while there's? Or is it a little?
Yeah, this okay, so first off, no, they did not ethically like take an embryo out. That would be ethical issues, but unfortunately, there are things like spontaneous miscarriages and things like that. So in many cases, what would happen is there has been some times where like there's been a miscarriage in the hospital. They've got the kind of aborted fetus. It's not going to make it. There's nothing they can do for it. And sometimes they do kind of donate those for scientific purposes and some research purposes occasionally. This is probably, unfortunately, the picture that I'm showing you here is an example of that. So there is also examples though of molecular homology. And molecular homology, this is similarities in molecules and genetics, for instance. So one example of molecular homology is that all living organisms use the same genetic or a language DNA and occasionally RNA for viruses, but it's actually debatable if viruses are true living organisms or not. But all cellular life uses DNA as genetic material. Not only that, we can go a little bit more specific. All cellular life uses the exact same genetic code. When I say genetic code, what do I mean? They are the codons that code for amino acids. So we did the genetic code, like for instance, the UUC codon codes for proline and or things like that. But every single form of cellular life uses the exact same genetic code. And this probably is very, very good evidence for common ancestry. Why does all genetic life use the exact same genetic code? It's probably because all cellular life can be traced back to a single common ancestor. That was the genetic code in that single common ancestor. And then all genetic life is derived from that common ancestor. That's a big piece of evidence for that kind of shared common ancestry theory. If we look at other things, we can also see gene sequences are often very, very similar between organisms that share a recent common ancestor or sometimes even distant common ancestors. So for instance, I could go through this, but I think I got my numbers slightly off. But if we share about 99% of our gene sequences with chimpanzees, which we can trace humans and chimpanzees back to a common ancestor from fossil records. So pretty similar. We share about 75% of our gene sequences with things like mice, which is another mammal. We only share probably about 40 to 50% of our gene sequences with then yeast, which is a single cell type of microorganism, but it's still a eukaryotic cell type, for instance. So we can take a look at these gene similarities and use those to determine ancestry. And we're going to talk more specifically about that later in the semester. Okay. But even if you look at very, very distant ancestors like bacteria versus humans, we're going to find some gene sequences in there. For instance, the gene sequences for things like DNA polymerases or RNA polymerases or even ribosomes. Those are really important enzymes for cellular life. Those share a lot of DNA sequence similarities. They're not exact copies, but they're going to be, we see some similarities in there in the coding sequences. Another example of homology as that where we can see evolution. We can use homology to see evidence of evolution is what is called vestigial structures. Vestigial structures are remnants of features that served an important function maybe in an ancestor, but now don't do anything. So they're remnants, they're non-functional versions of something that don't work in us currently, but probably worked in a common ancestor and did something for a common ancestor. And Darwin provided several different examples of vestigial structures and the origin of species. But there's a lot of different versions of this. So cave fish, cave salamanders are blind. They live in the dark all the time, sorry. It was actually I could leave it on because one time I missed a call from my kids' school when they were sick. And that was actually a call from my kids' school, but then I saw the email come in simultaneously that they're testing their new phone system. Anyways, all right. So what ended up happening here is that the cavefish are blind, salamanders are blind. They live in the dark all the time. Eyes don't do them any good. But their ancestors of those fish and the salamanders, they were some type of animal that moved in from outside the cave into those cave ecosystems. So their ancestors had eyes. But those cave fish and those salamanders have stayed, and those populations have stayed in the caves for so long. It actually turns out while they have vestigial eyes, their eyes do not work whatsoever and are blind. The kind of connections have been kind of missed, and so they have remnants of eyes, but they don't work. And that's because eyes are expensive. Eyes that cost a lot of energy to use our eyes. So in these things, these cavefish and cave salamanders, it was expensive for them to still kind of be using the eyes, even though they gave them no advantage whatsoever. So mutations happened that inactivated the eyes and lowered the energy cost. But they could not get rid of the eyes altogether. It's a historical constraint, so they got left there, for instance. There's vestigial structures for pelvis and leg bones and snakes. The ancestor of snakes had legs. And they lost legs over time, but you still see evidence in their skeleton of pelvis and those leg bones in there. Tailbones in humans, again, humans don't have tails, but we kind of have a tailbone where a tail would have been. And that's still left over there. Goosebumps is a vestigial response. Anybody ever get goosebumps? You get the bumps on your skin and then your hair kind of stands up. Well, that's a leftover response from our ancestors. Our ancestors had more fur, more hair across their body. And when they were cold, they would get goosebumps and cause their hair to stick up. But what would end up happening is because their hair would be sticking up, it would trap a layer of air between their skin and their fur. And that was an insulating area. It was insulating air to help keep them warmer. So the goosebump response, you get goosebumps because you're cold, because it's trying to do a response to keep you warmer, but that only worked in our ancestors and it doesn't work for us anymore. But it's a vestigial response then. And it's one example you guys might get of vestigial structures is appendix as an example, and actually they think it actually does stuff now. So the appendix is commonly given as an example of vestigial structure that probably did something in the past, but it doesn't do anything anymore. They actually think it does do stuff. So for instance, if you have a GI tract disorder, the appendix is a little pouch that comes off of your large intestines. What ends up happening here is they thought it wasn't really doing anything and it was left over from something that did something in an ancestor. What they actually think is if you have a GI tract disorder, it actually can cause all the kind of good bacterial populations in your gut to deplete. And then the appendix, as a little pouch, acts as a store of your kind of good bacteria and good microflora. And then following a GI tract disorder where you have like diarrhea, dysentery, things like that, it actually creates a little colony of bacteria to repopulate your gut and help you get better faster, for instance. So it seems like the appendix is more developed in certain countries where sanitation is more of an issue. Traits and homology can be used to classify organisms. So this is an evolutionary tree. We're going to be going through evolutionary tree and taxonomic trees later in the semester. But what can happen is we can sometimes use molecular DNA or homology of DNA to actually put together, and homology traits to put together things. Like for instance, it basically can be used as traits and homology to classify organisms, like the development of the amnion during embryonic development. This is a amniotic sac during the things. We find that in mammals, lizard snakes, crocodiles, ostriches, hawks, and birds. So we might think these are more related to one another in terms of their evolutionary history, but amphibians and fish don't have amnions. So they're more distantly related. But this tree is showing relatedness. The shorter the branches, the more related they are to one another, for instance. But you can have different types of traits being developed, and they use those traits to kind of see how they might be related to one another. Again, homologous structures are due to common ancestry. The ancestor had a structure. There might have been some speciation events. And then the descendants of that common ancestor and those descendant species then have a similar but modified version of that structure. That's kind of the idea with homology. However, you can sometimes have trait similarity without shared ancestry. So analogous structures, these are resemblances independent of shared ancestry, without close relationships. And they are due instead to something called convergent evolution. And convergent evolution, this is where similar selective pressures result in the evolution of similar adaptations or features. Basically, same trait evolves independently multiple times. Well, 2 or more times is another way for me to say that. So if I'm just kind of looking at this, if I'm looking at homology, you might have a version of a trait in some type of an ancestor. Maybe it undergoes speciation events. So this might be some descendant species, for instance. Okay, so this might be the ancestor, these might be the descendants. With homology, trait similarity due to shared ancestry, you have maybe trait number 1, you have a version of it, but maybe you have a trait number 1A, 1B, 1C, 1D. You have that trait, but a modified version of that trait. Does that make sense, everybody? In analogous structures, you might go through this tree again. But the idea with then something like a analogous structure is that it's a trait similarity without not due to shared ancestry. So maybe you have a evolution event here and you get a version of trait number 1. Maybe you have an evolutionary event here that is a similar selective pressure produces a version of that trait trait as well. So you have that version of trait 1 here and here on those branches. But you're missing it here and here. And that is because also that trait did not exist in the ancestor either. Okay, so that's kind of what we're talking about with this version. And what you had to see is you had to have 2 different evolutionary events, one on this branch and one on this branch that produced that trait independent of one another. So this would be kind of basically my way of kind of working through what is the difference between homology and homologous structures versus analogy or analogous structures. Questions about that? An example of convergent evolution is sugar gliders versus flying squirrels. They have webbing between their front and their back legs that allows them to glide long distances through the air from tree to tree. You find flying squirrels in the Western US. You find sugar gliders in Australia. And they're similar in many ways. They have this similar gliding mechanism. But it turns out the ancestors of the sugar gliders could not glide. The ancestors of the flying squirrels could not glide. Okay? They evolved that ability over time, but they evolved it after Australia was all the way over here and North America was all the way over here. Okay, so in North America and Australia are obviously not connected. They're separated by the entirety of the Pacific Ocean. There's no interbreeding or mixing between these 2 populations. And while they were separated, they both independently evolved this gliding mechanism due to some similar selective pressures. They both grow in some temperate forests type of environments. Another example of convergent evolution is glass lizards. Were thought to be snakes. They don't have legs, but it turns out they're not snakes. They look like snakes. They act like snakes, but it turns out they're lizards. Technically, they're just limbless lizards because snakes evolved way before glass lizards, and glass lizards really just lost their legs very, very, very recently from an evolutionary standpoint.
Wings and birds and bats.
Wings of birds and bats would be another example, of convergent evolution. Yep. Different ways, independently evolved ways to fly. All right, another evidence for evolution we're going to talk about today is biogeography. And this was a major line of evidence that Darwin used in The Origin of Species. But biogeography just describes the geographic distribution of species. So life and geography stuck together. but it's where you find living organisms, living species out in the environment from a geographical standpoint. Looks at the distribution. Biogeography in particular has been useful when studying islands. Islands are a big thing that's been used to study evolution. This is because islands have many what are called endemic species. And an endemic species is a species that's found nowhere else in the world. So islands tend to have a lot of endemic species. You find that species on that island, but only on that island. And also these species, while it is their own species, they tend to be closely related to species on the nearest mainland island. Or the nearest mainland or the nearest next island, I should say. Darwin explained this by suggesting that islands are colonized by nearby species. And then those species, those founding populations give rise to new species as they adapt to their new surroundings in a process called adaptive radiation. And adaptive radiation basically just means something along the lines is they hop and then they change. They hop to a new spot and then they change some more. But this basically, the idea here is as they get these moving populations, they adapt to the new surroundings and they change to adapt to those new surroundings. Okay, and this is called adaptive radiation because they're radiating or moving and then adapting. This is actually, again, islands cause this quite a bit. I use the example from actually just the quiz on this then as well to kind of start hitting this idea. But what can happen is when you have, say, a big mainland population, some of the members of the population break off, but this creates what's called the founder effect. When they move to that new location, maybe an island out here, okay? That means they have that gene pool of that founding population is often different than the mainland, so they've already changed right from the get-go. It's usually a small population. What happens to small populations? Genetic drift, which can cause some random allele fluctuations in that population. But also the island, do you think the island has the exact same environmental and selective pressures as a mainland continent? Nope. So you're also going to have different selective pressures favoring different traits. So you can have founder effect affecting that founding population, you can have drift affecting them, you can have natural selection affecting them. And all of that can simultaneously be causing change, and pretty substantial change in a fairly short time frame as well. Questions about that? So yeah, they're going to be subject to founder effect, they're going to be subject to drift, they're going to be subject to natural selection. And even could be mutations, all of that happening in the background then as well. And just as an example of this, Hawaii is a well-studied example of all of this. There is adaptive radiation of plant species. So there is this plant in the Dubatia genus, and you can actually see it hopping from island. It's basically moved from island to island in Hawaii over time. And it actually, while all of these look quite different, they are all within a very closely related genus of plants that have undergone some modifications and some changes, especially because these different islands have different habitats. The big island of Hawaii, for instance, is the oldest one, and it has got a kind of well-established jungles and forests and things like that. Some of the islands over here are younger and newer, and so they actually are more volcanic in terms of the soil composition, and you get different types of plants living in those areas. All right. Darwin also noted that ocean islands lacked terrestrial mammals and amphibians like frogs, but had a bunch of flying animals or flying mammals in many cases. Why? Why can't frogs get to islands in the ocean? Well, they can. Salt water kills them. Frogs are just fine in fresh water, but if you put a frog in salt water and ocean water, it's going to kill it. So you just don't get frogs or a lot of other types of amphibian species on ocean islands. The salt water is just too bad for them, for instance. But he took this as, and the fact that flying things are often on islands is just because, well, they can fly. over the ocean and reach the islands more easily than other stuff. And so he also took this as evidence for the colonization theory then as well for biogeography. All right, finishing up biogeography, I know I'm kind of close to time here. Earth's continents, you guys probably have all heard of Pangea before. One big supercontinent. If you actually look at Pangea, you can see how it broke apart into what are now the modern day continents over time. But for instance, you've seen the things like South America used to be directly joined onto Africa. And by the way, even nowadays, you can see how they kind of come together like puzzle pieces. But what that's done is our understanding of continental movement has allowed us to predict things like fossil locations. So during dinosaur times, South America and Africa were joined together. And in fact, we find fossils of dinosaurs in South America and Africa that are the same species of dinosaur. Even though they're on different continents now, what we found is that they used to be together in the supercontinents. And then those continents drifted apart, those fossils still stay there, and we can see the same species of dinosaur on separate continents. And we can know that because, well, they didn't used to be separated. And it used to be just one population of dinosaurs in one area, that for instance. Questions about that? That's one example, but we've seen other things like, for instance, Alaska used to be at the equator. And it had rainforests. And all those rainforests over time, the plant material died, got buried in the ground, got pressed and heated, and it turned into oil. And that's why there's lots of oil deposits in Alaska, for instance. Because it used to be rainforests and all that plant material got buried in the ground, and all that carbon-based plant material gets converted into oil. That's why you find oil in Alaska, and that actually allows us to also predict where oil deposits might be current day, because they're not always in the spots you would predict. What about that? All right, we'll pick up there talking about fossil record on Monday, but we're also going to start talking about then population genetics on Monday.